Okay guys, so if we look at the structure of a chromosome, so exactly what the chromosome is, remember your chromosome is made up of all your DNA. Okay, so humans have 46 chromosomes divided into 23 pairs, and that has all of our DNA arranged in specific bases. Okay, now that's a lot of bases. There's millions and millions and millions. So what happens is those DNA bases that you have all coil up together into one big lump that you can see, okay, as a dark mass. And if we simplify that, it will be shown kind of like that. And we call each one of those DNA strands a chromatid. Okay, so remember our chromatid is going to be our DNA strand. Now, when we want to multiply our cells, we need to replicate the DNA. So the first thing that happens is DNA replication. And we already know what happens in DNA replication and the stages. So this one chromatid is going to make an exact copy of itself, and it's going to make another chromatid. Okay, so this side is going to be an identical chromatid of DNA. That's been replicated. Okay, now this whole thing is going to be my chromosome. So it's made up of two chromatids, and in the middle, it's held together by what we call a centromere. So a centromere holds the chromatids together, and it's just a section where bases don't code for anything. Okay. Now, along each part of that chromatid, it's divided up into little sections of DNA. So we can have this whole section of DNA here, which is going to be the same as this whole section on the DNA here. Then there's going to be another section underneath it, which is going to be different. And another section, okay, all the way down to the bottom. And all of those represent our genes. So each section of DNA there is a gene, and each gene codes for a different protein. Okay, so we have our genes going all the way through all of our different chromosomes. And then at the very end of each one, there's a little section at the top and at the bottom of the exact same section of DNA. Now those little sections at the top and the bottom we call telomeres. Now what a telomere is, it's a repeated sequence of DNA where there's no information coding for a gene. All it is a simple sequence repeated over and over and over again, so that when we replicate the DNA and our enzyme gets to the end and it breaks off, that no important information is lost, because each time that enzyme gets to the end and it breaks off, it loses a few little bases. So every time we replicate the DNA, we lose a few bases, and we lose a few more, and a few more, etc. So those telomeres get smaller and smaller and smaller each time the DNA is replicated, until there is no more telomere left. And when the telomere is gone, that means the cell must die, because now it cannot replicate anymore, and that then indicates that it's time for that cell to die. Okay, so telomeres are super important, even though they don't actually code for anything. Okay, and now this is going to be one chromosome, And remember, each chromosome has another one that is pretty much almost identical, okay, because you have pairs. So this one, same shape, same size, everything, same number of bases, same genes on it, section-wise. But this one comes from your mother and this one comes from your father, because remember, we have 23 pairs of chromosomes. So when we have them paired up, okay, we call those pairs homologous. So we have 23 homologous pairs of chromosomes. So 46 chromosomes in total, divided up into homologous pairs, meaning that they match the same. You just got one from your mother and one from your father. And then you read the specific genes in them. Okay, so that is what a chromosome looks like. Don't forget all the different terminology that I just explained. So then we can move on to mitosis next.